How does a cell copy its DNA? The process of DNA replication occurs. Here we see two DNA chains. Notice 5' to 3', and then the reverse 3' to 5'. Where do these 5' to 3' come from? Well, they actually come from the positions of the carbon on the 5 carbon component of the nucleotide, 5 carbon sugar and 3 carbon sugar. Remember, DNA is a double helix, two chains that are anti-parallel. So on this top chain, the 5 carbon position on the 5 carbon sugar adds 5 to the third position of the carbon on the next nucleotide, 5, 3, 5, 3, and so forth. On the opposite chain, which runs in a different direction, this is reversed. So, how do we replicate DNA? We first use an enzyme called helicase to unwind the DNA double helix. Single strand binding proteins help keep the chains unwind. DNA has a tendency to come back together to form the double helix. The most important enzyme, DNA polymerase, adds nucleotides to form the new chain. However, DNA polymerase can only add nucleotides in the 5' to 3' direction. Hence, it forms a continuous new DNA strand. What that means is that this strand is the leading strand. Now, notice in the opposite template strand, notice how primase will form a short RNA primer. This then allows DNA polymerase to add nucleotides, but then it forms short pieces or short segments, or even specifically short Okazaki fragments. Well, because it is discontinuous compared to the continuous formation on the leading strand, this strand with the Okazaki fragments is called the lagging strand. Now you have these Okazaki fragments, but then DNA ligase, another enzyme, combines these fragments together to eventually form another DNA double helix. So, what do you notice? Notice that you have an original strand from the original DNA double helix. So, we call this DNA replication as semi-conservative because you maintain one template from the original strand and then you form a new strand. Similarly, on the lagging strand, the same thing eventually forms.